Hello, everyone, and welcome to today's broadcast, Lung Cancer Evolution and Immune Escape. It is presented by Nicholas McGranahan, PhD, a Senior Research Associate at the Cancer Genome Evolution Research Group at University College London. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before I begin, I'd like to remind everyone this event is interactive, and we encourage you to participate by submitting as many questions as you want, anytime you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. McGranahan. I will now turn the presentation over to him.
Excellent. Thank you very much for the introduction, and I'm very excited to tell you about some of the work we've been doing on lung cancer evolution and immune escape. So just to begin with my disclosures, I have received some consultancy fees from Achilles Therapeutics, so then that brings me to the actual start of the talk. And to begin with, I thought I'd just introduce one of the concepts that really fascinates us, and this is really the focus of my lab, is the concept of heterogeneity in cancer. And this heterogeneity can occur on many different levels. First of all, there's inter-tumour heterogeneity, in that there's no two tumours are identical. And clearly this makes sense in terms of a breast tumour being different from a lung tumour, and the histological subtype of the tumours can be very different. But even within a single tumour type, there can be striking diversity. And this indeed is what's given rise to the notion of precision medicine, that based on the person's individual tumour, you might be able to guide a specific treatment. But even within a single tumour, there can also exist diversity. And this is what we call intra-tumour heterogeneity, depicted here as multiple competing or cooperating subclones. And this diversity potentially can act as the substrate for Darwinian tumor evolution, and therefore may lead to treatment resistance. And the focus of this talk will be very much on how extensive is this diversity within tumors, and how can we use this diversity to understand how a tumor has evolved, and what is the interaction and the relevance between this diversity and cancer immunity. And most of what I'm going to tell you about today is in the context of lung cancer. So this leads to the question, so why study lung cancer? Well, unfortunately, I think the numbers really speak for themselves. As you can see in this plot here on the right, the incidence of lung cancer really is staggeringly high, and unfortunately so is the mortality rate. There's approximately 1.4 million deaths worldwide from lung cancer, and really at the moment, despite surgery and adjuvant chemotherapy, the survival rates really are quite dismal, with 5-year overall survival rate less than 10%. So I think there's a pressing need to understand why the survival rate is so poor, and to fully understand what might be driving this resistance to treatment and the poor prognosis that we observe. So most of what I'm going to tell you is in the context of the TRACER-X study, which is a study that's been set up to track non-small cell lung cancer evolution through therapy, looking particularly to start with the early stage tumours and explore really what is the evolution of these tumours, and what is the relevance of intra-tumour heterogeneity. So to give you a bit more background, I can show you how do we explore tumour evolution and intra-tumour heterogeneity. Well, the first thing we do is, rather than take a single sample of each tumour, we take multiple regions of each tumour, here depicted in this diagram. So these are tumours that were removed with surgery with curative intent. Then we take multiple tumour regions, and each one of these tumour regions is then subject to whole exome or whole genome sequencing. We also sequence the matched germline. And using this, we can therefore decipher what are the specific somatic mutations present within each tumor region, and how do these differ between the tumor regions. And then integrating this information, we can derive a phylogenetic tree of how the tumors evolved. So this allows us to determine what are the early clonal mutations, those on the trunk of the tumor's phylogenetic tree that are present in every cancer cell. And then also what are the subclonal mutations, those only found in a subset of the tumor regions and also a subset of the cancer cells within the entire tumor. So that's how we do it. So then the question is, how extensive is this heterogeneity? Well, what I'm showing you here is the first 100 patients that we've sequenced within the TracerX study. And here, every column is one of the patient's tumors. And if it's blue, that means it's a clonal mutation. So by clonal, I mean that we think that this mutation is present. In red, these are subclonal mutations found only in a subset of the cancer cells. And what you can see here is there's certain patients' tumor regions we think for certain tumors here have thousands of subclonal mutations. So on the far left, you can see certain tumors that the vast majority of the mutations are subclonals, found only in a subset of the cancer cells. Whereas there's other tumors where the vast majority are clonals. There's really also here we see differences in the heterogeneity of heterogeneity itself, with clear differences between tumors and also between lung adenocarcinomas and lung squamous cell carcinomas. So this is the simplest way of looking at heterogeneity, just in these two categories. The next question we can ask is there actually more information within a single region? Well, one thing we can do is we can actually look at each mutation here, and I'm showing you here a schematic of how we can do this. If we look at this case, we sequence 4 regions, we can integrate the variant allele frequencies that we get from the sequencing reads with the purity and ploidy. And this allows us to derive an estimate of the cancer cell fraction. So that's the fraction of cancer cells that harbor that given mutation. And then as we have an estimate in each tumor region, we can then cluster these to try to obtain the actual subclones that exist or have existed during a tumor's evolution. And then looking at the various prevalence of these, using various techniques, we can actually start to resolve their relation to each other, ultimately to get a phylogenetic tree and a regional subclonal architecture. So we can actually look, so if we go to the next slide, what you can actually see here is an example here where we've got 4 tumor regions. And in each tumor region, we can look at the subclonal makeup of the subclones that exist within each tumor region. This allows you to see, for instance, that region 2 and region 4, they share a lot of the same subclones, whereas region 1 and region 3 are actually quite distinct, both from each other and from the other tumor regions. So we can do this in one tumor, and then we can also do this across the whole cohort now. So we can start to look at the forest of evolutionary trees of how each tumor has evolved. And this allows us to understand that while certain tumor regions or certain tumors are actually fairly simple in their evolutionary histories, others are remarkably complex. So this is looking at the mutational level, and as I said, first of all, the clonal and subclonal mutations. We can also consider this at the copy number level. We do this here is looking at what we'd expect across chromosomes 1 to 22. If it's a normal cell, we'd expect 2 copies of every chromosome, 1 maternal and 1 paternal. But what we actually see in these tumors is something quite striking. So if you look here in region 1, rather than 2 copies across the genome, we see the vast majority of the genome is present 4 copies, suggesting there's been a whole genome doubling event at some point during the tumor's evolutionary history. And again, because we've done the multi-region sequencing here, we can look at this one region and we can also compare that to the other tumor regions. Again, we see overall quite a similar profile, but there are also certain events that are specific to individual tumor regions, again, suggesting there's copy number heterogeneity and diversity within tumors at the copy number level. So now we can add this to our plot to look at mutations as well as copy number events. And then we can ask the question, is this clinically relevant? Does the amount of diversity within a tumor, does that predict response to treatment or treatment failure? And this is really one of the key questions we wanted to ask for the TracerX study. And so to do this, we initially said, let's do this in the simplest way possible. Let's just take at the mutational level and look at the proportion of heterogeneous mutations and split that according to the median. When we did that, we saw actually we didn't see any clear difference in the survival curves when we split according to the medium of intra-tumour heterogeneity, the mutational level. But interestingly, when we did the same comparison at the copy number level, looking at copy number ITH, we saw quite a striking difference in the survival curves. So those patients with very homogeneous tumours at the copy number level, where all the copy number events themselves were homogeneous, seemed to do far better than patients where the tumours are with lots of copy number heterogeneity, which to us suggests that perhaps this indicates that ongoing chromosomal instability and gains of losses of chromosomes within separate subclones is very prognostic. And indeed, when we look and compare copy number ITH compared to a static measure of how much copy number aberrations the tumour has, we see it's the copy number ITH here that's particularly prognostic, strongly suggesting that it's this ongoing dynamic instability which really fosters a poor outcome here for these patients. And we find this is significant in multivariate analysis as well. So this ultimately leads to the questions, what drives this heterogeneity that we're seeing at the copy number level and at the mutational level? So next we wanted to look in a bit more depth, particularly this chromosomal instability. Can we go beyond this simple classification? Well, one thing we reasoned is we can actually look at the maternal and paternal SNPs that are present across the genome. This is work primarily by Tom Watkins in the lab. So here what we reasoned is we have maternal and paternal chromosomes, and when we have a copy number event, we can actually see differences in the prevalence of the maternal and paternal chromosomes. And the way we do this is we look at the SNPs, the single nucleotide polymorphisms, and we look at those specifically that differ between the maternal and paternal chromosomes, those heterozygous SNPs, as we call them. And here we can see in the normal, they're present at 50%, because we said we've got one copy from your mother and one copy from your father. But when you have a copy number event, what you see is you see a separation of these SNPs, because now rather than present at 50%, here you see the paternal one is present at 2/3, and the maternal is present at 1/3. So these two bands separate here in region 2. So here, this is actually one of the main ways that copy number tools work to try to decipher copy number events. But if we also have a copy number event in tumor region 1, we can see we also see separation of these bands. And if we were to apply a standard tool, we might say tumor region 1 and tumor region 2 look very similar. But actually now if we're looking at the orange and blue, we can see there's a difference here because one is a maternal gain and the other is a paternal gain. So what we can do here is we can phase these SNPs. So what we say is, let's take, for region 1, let's take all the SNPs in the upper band, let's label them as orange, and all the SNPs in the lower band, let's simply label them blue. And then we can apply that same phasing to the second tumor region, and now what we can actually distinguish and say that these are clearly separate events that are occurring in parallel during tumor evolution. And we call this mirrored subclonal allelic imbalance. And just to show you a bit more depth, we could actually look at this across one patient's tumor here. And this is a particular patient where, again, we see separation of the bands. And here we could label again with orange and blue. And in this case, we saw actually over 45% of the genome here, looking at chromosomes 1 to 22. We found this mirrored subclonal allelic imbalance. And here in particular, I've highlighted here on chromosome 14, where you can see the orange is on top in certain regions, whereas in other regions, we have the blue on top. And overall, when we look across the TRACERx cohort, these 100 patients that we've started looking at in more depth, we see in the majority of patients, we see this mirrored subclonal allelic imbalance, which again is highly consistent with chromosomal instability that's ongoing during tumor evolution. So this suggests there's ongoing events, but are these actually important? Are these actually selected for during tumor evolution? Well, one thing we can look at is if we have independent gains of oncogenes, so key cancer genes. When we do this, we actually saw, we got the tumor's phylogenetic tree, we saw the same genes being amplified on different alleles on different branches of the tumor's phylogenetic tree. So this suggests there's convergent evolution here in terms of the same events being selected independently across in different subclones. So this strongly suggests there's selection that's ongoing here, because the chance of this happening, just purely the probability of this happening by chance is incredibly slim. So we see here one example, and we could look further across the cohort, and we see these key amplification events actually happening in 5 tumors. In one case, we actually saw 3 independent events of the CDK4 amplification, suggesting there's a strong selection pressure and constraints here for the tumour evolution that it really needs to potentially gain the CDK4 amplification. Potentially, this may guide treatment strategies as well. And overall, we see actually this parallel evolution, which is indicative selection in 17 of these tumours. So we think this copy number heterogeneity really might be indicating dynamic ongoing chromosomal instability, which permits selection for key events occurring at the copy number level. So next, I'd like to talk a bit more about other drivers of ITH and link to this one that we see that is related to copy number heterogeneity, which is genome doubling. Okay, so genome doubling has actually been quite well described in cancers, and particularly in lung cancers. You can use sequencing data to look for whole genome doubling events, and lung cancers this appear to occur in approximately 60% of tumours. So this leads to the question is actually what happens when you double your genome. This is something we started to explore a few years ago, looking actually in a colorectal cancer cell line, HCT116. And when you look at the DNA content of this cell line, you can actually see there's a naturally occurring population of cells that are present at greater than 4N, i.e. tetraploid genome doubled cells. So we wanted to ask, okay, well, can we isolate these cells and grow these in culture and see, are there any changes? Is there any association with chromosomal instability when you double your genome? And so this is exactly what Sally did. She grew these cells and she obtained essentially a family tree of genome doubled here in red and non-genome doubled clones in blue, where we can look at the different genome doubled clones and compare them to their sister non-genome doubled clones. And when we looked at these in long-term culture, so we grew these for almost 2 years in culture, we could actually see, are there any changes that accumulate over time? And here, looking at the non-genome doubled clones, so what I'm showing you here is passage 5 through to 75. And again, I've got chromosomes here on the y-axis, chromosomes 1 to 22. A copy number gain is in red, and a copy number loss is in blue. And what you can see in these non-genome double clones, it's really quite a boring picture. There's certain gains that were present in the original founder clone HCT116, but then these just remain present, and there are no additional gains and losses, which is interesting because HCT116 is making segregation errors. We can see that, but they don't seem to tolerate these errors. And when we look at the genome double clones, quite a different picture emerges. So here again, we're looking at passage 5 through to 75, where 75 is almost 2 years in culture. And here, rather than it just being those original gains events, we can actually see we're accumulating a lot of loss events. So that's from the doubled cells. Once they've doubled, they start to lose parts of chromosomes. And in particular, one thing you can see here is a region of chromosome 4 is lost in the end in every clone that we've sequenced. And so these losses are occurring again independently, suggesting potentially there's some sort of selection pressure or fitness advantage to losing this particular region of chromosome 4. So this allows us now to compare these clones that were grown in culture to what we actually see in the tumours. And here, what I'm showing you here on the y-axis is a measure of chromosomal instability based on the proportion of the genome that differs from ploidy. What we can see is when we compare chromosomal instability to ploidy, there's 3 general populations. There's the diploid tumours, then there's the tetraploid tumours, and then there's these triploid tumours that seem to have slightly higher levels of this chromosomal instability score. And when we look at our clones over time, these genome double clones, we can see that they gradually become more unstable in terms of they increase in chromosomal instability, but they also drift down in terms of ploidy. So they become more triploid, suggesting perhaps there's an early genome doubling event, and then they become more and more unstable. So overall, we can say that perhaps genome doubling appears to be permissive for the tolerance of chromosomal instability. And after a genome doubling event, there seems to be this rapid cancer genome evolution. And so this leads to the question is perhaps genome doubling could act as some sort of warning for early impending chromosomal instability. So perhaps this is associated with poor prognosis. And when we looked at this in colorectal cancer here, and using the Cancer Genome Atlas cohort, we did see a separation of the survival curves between those tumours that undergone a genome doubling event in red that hadn't in blue. And we could also validate this in an independent cohort. And quite reassuringly now, more recent data looking in more later stage tumours have found across pan-cancer and across different cancer types here, that we see again an association between genome doubling and survival, with those tumours that have undergone a whole genome doubling event appearing to do, patients unfortunately appear to do far worse than their non-doubled counterparts. And when we go back to the TRACERx data and the lung data, this also fits with that. When we see a genome doubling event here, particularly in lung adenocarcinoma, we see an increase in heterogeneity, both at the mutational level and critically also at the copy number level. So we think this genome doubling event is perhaps a critical event that permits accelerated cancer genome evolution. And we do see this frequently and often as a clonal event in lung cancer evolution. So we've focused so far on copy number events, chromosome instability and genome doubling. And finally, I'd like to also consider some of the mutational events and what drives later events during tumor evolution here. Well, one thing we can look at is we can look at the different mutation types that exist. So ultimately, if we're just looking at single base substitutions, then there's 6 different types we can consider, because there's 4 different nucleotide substitution types. These can change into 3 others. That means there's 12. But in general, we can't necessarily tell what strand of DNA it occurs on. So this reduces down to 6 substitution types, depicted here as 6 different colors. And the different prevalence of this substitution types can give us hints as to what's caused the mutation. And quite well established here is actually that smoking is associated with C to A transversions, which in this plot here is depicted as this gray part of the pie. And when we looked at this, we looked a few years ago actually in patients, and we looked here, patient 11 was a very heavy smoker that smoked a packet and a half of cigarettes a day for over 30 years. We saw lots of these C to A transversions, which made sense to us because patient smoked a lot. Patient 1, on the other hand, was an ex-smoker, and we saw slightly fewer C to A transversions. And patient 3 here was a never smoker, and we saw even fewer. So this made sense, and indeed this was just confirming what had already been seen in the literature. But with the multi-region sequencing, as I've tried to demonstrate, we can also start to look at the tumour's phylogenetic tree. And this allows us to say, do we see differences between the early events, those on the trunk? Do we see any difference between those in the later events, those on the branches? And here, when we looked in patient 8 at the early mutations, we saw lots of C to A transversions. And this patient was an ex-smoker. So this suggests these mutations happened while the patient was smoking. And indeed, when we looked at the later mutations, we saw far fewer C to A transversions. This is what we intuitively expected because this patient had stopped smoking. So we could look overall in this cohort of ex-smokers and current smokers, and we saw the early mutations were dominated by C to A transversions. So even in the ex-smokers, most of the early mutations were caused potentially by smoking. And then we could also look at the later mutations. And in this case, we saw that in the ex-smokers, we expected a reduction in the C to A transversions. But interestingly, in the current smokers, we also saw a reduction in C to A transversions. So these tumours, despite the continuous mutagenic insults of tobacco smoke, it seemed that the proportion of mutations caused by smoking was going down. So this suggests perhaps there's an additional mutational process taking over here in these tumours. And indeed, here we saw an increase of C to G and C to T mutations. But what's actually causing this increase? Well, here what we did is, rather than look at just these 6 substitution types, we can actually consider the nucleotide either side of the mutated base. So here, rather than just 1, we can consider 16 different types of C to A transversions. And we can do this look now at each of the 3 substitution types that were really changing here. And when we do that, that means it's 48 different substitution types. Let's just go there. 48 different substitution types. And here, when we now look at the early mutations, we can consider these 48 types. And we can see that the early ones here, again, this is a patient that was an ex-smoker. We can see the early mutations were dominated by these grey bars, and they're relatively fat across these 16 different types of C mutations. But then when we looked at the later mutations, we saw quite a striking difference. You see the mutations here, particularly at the TPC sites. So then we asked, what's actually causing those mutations? Well, this profile of mutations is very similar to those caused by APOBEC an enzyme involved in innate immunity, in particular AFBEC3B. So these TPC mutations we think are caused by potentially AFBEC3B enzyme that becomes active and mutates DNA. In particular, it's thought to mutate single-stranded DNA. So here what we think is the early mutations are caused by smoking, and then while they still cause late mutations, the current smokers, AFBEC often takes over as a dominant mutational force. So this suggests we're getting a difference in mutational processes over time, but we can also look in space. And in one tumor here, we had regions from a squamous cell carcinoma and a region from an adenocarcinoma. And here, when you look at, again, the trinucleotide context, you can see that we see AFBEC, particularly in the adenocarcinoma region, but not in the squamous cell carcinoma region. So this is just in a small cohort. We could then look across the TRACERx cohort, and actually we see a remarkably similar picture, with the early mutations being dominated by tobacco smoke on the whole. This is restricted to those patients that are current or ex-smokers, where the later mutations saw less tobacco smoke and more signature to AFBEC mutations and also other mutational signatures taking over, suggesting that although tobacco smoke causes a lot of mutations, there's often additional processes that are ongoing during cancer evolution in lungs. Okay, so this allows us to start to build up a model of how we think non-small cell lung cancers evolve. So I'd just like to briefly go through this. So first of all, and I haven't gone into detail about this, we think there's certain mutations that frequently occur early during tumor evolution. In terms of cancer genes that are mutated, in particular, in both subtypes here, we see p53 mutations being some of the earliest events. And then frequently we have a genome doubling event. This allows us to also time mutations that occur before the doubling and those that occur after the doubling event, because those that occur before will be present at 2 copies, because they will have been doubled as a result of the genome doubling. So then we can see there's other cancer genes that more frequently occur early. In this case, we see, for instance, PIK3CA mutations frequently occur later in lung adenocarcinomas. And then we also see even later mutations, and these are often mutations in genes involved in DNA repair and also chromatin modification. Another thing we can also look at, which I think is quite interesting here, is that when we look at the different signatures that occur, we can see in the current smokers, we see the smoking signature both before and after genome doubling. But interestingly, we also see the smoking signature in lung adenocarcinoma, both before and after genome doubling in long-term ex-smokers. Now, these patients, many of these long-term ex-smokers stopped smoking 15 years ago, which suggests this genome doubling event, we can place that when the patient was still smoking. So that means these tumors have potentially been developing for a very long period of time. And we see this in lung adenocarcinoma, but not lung squamous cell carcinoma, suggesting potentially different modes of tumor evolution here in these different subtypes. Okay, so for the next part of the talk, I'd like to focus a bit more about the interaction between a cancer cell and the immune system. Focus not just on the genomics, but also on how that might be recognized by the immune system. So how do we do this? Well, computationally, what we can look at is we can look at whether mutations might give rise to peptides that are presented by the patient's own HLA molecules and therefore recognized by CD8 T cells. And these peptides may arise from mutations that are tumor-specific. And these therefore are highly patient-specific. And the way computationally we check for these, what we call neoantigens, is to check whether the peptides that contain the mutation, whether these bind to the patient's own HLA molecules. And we use NetMHC for this. So we can predict for every patient a set of potential neoantigens. And we can next ask, are these actually important? Well, this we initially looked at by looking in the Cancer Genome Atlas in lung adenocarcinoma patients. Here we had 150 cases. Did mutation calling, we did HLA typing, we also did neoantigen prediction. And here we predicted not just neoantigens, but whether these were clonal or subclonal, so whether these were found in every cancer cell or only in a subset of the cancer cells within the patient's tumor. And you can see here the majority of neoantigens we predict, we think are clonal, found in every cancer cell. And then we could ask, is this clinically relevant? So first of all, we just looked at all neoantigens. So when we split according to high and low here by the upper quartile versus the other quartiles, we saw those patients with a high burden seem to do somewhat better than those with a lower burden. And we could also look at heterogeneity here, where we split according to some measure of heterogeneity, we saw some separation of the survival curves. But then we could also combine these two and look not just at neoantigens, but clonal neoantigens, so those found in every cancer cell. Here we found patients that had a high burden of clonal neoantigens and low levels of intra-tumour heterogeneity seemed to do far better than those with a low level of low number of neoantigens or high heterogeneity, suggesting that you need to consider not just the number of neoantigens, but whether these neoantigens are clonal or subclonal. And this may more effectively elicit an immune response. But this is in the context of patients that were looking at primary tumour, and they won't necessarily have received any therapy relating to the immune system. So we next wanted to ask, well, what about in patients that receive immune checkpoint blockade, where these neoantigens are thought to be particularly important? And here, indeed, there's already been some work demonstrating that patients with a high burden of neoantigens in their tumour seem to derive a better clinical benefit than those patients with a low neoantigen burden. So this is data here that was published, very elegant data from Timothy Chan's group, was published in Science. So we said, okay, well, let's apply our computational pipeline and see if we get the same results. When we did this, we initially predicted the number of neoantigens per tumour, and then we could split these to those with a durable benefit and those patients with a non-durable benefit. And reassuringly, we saw results that were very similar to those that were published. So those patients with a high neoantigen burden seemed to do better than those with a low neoantigen burden. But what you can also see from this plot is there are a number of key outliers. So patients with a high number of predicted neoantigens that don't derive a benefit from immunotherapy. So we want to ask, well, can we add the clonality to this, and does that improve this association? Well, interestingly, when we did this, we could see that some of these outliers actually seem to have a very high subclonal burden, which again fits with the notion that not just the number of neoantigens, but their clonality matters. And indeed, when we looked at the measure of neoantigen ITH, we saw a clear difference between those that benefited and those that didn't. And overall, when we added this into our measure of prediction of whether a patient will benefit or not, we saw that improved the survival curves, the separation here. So when we looked at the survival in melanoma patients, we could also see when we separated according to number of neoantigens, we could see a separation of the survival curves, but this was again improved by incorporating a measure of ITH. And interestingly, when we looked in more detail in Eli Van Allen's cohort published with Levi Garraway, we could see again a separation of the responders and non-responders. There were certain non-responders with a really high level of subclonal mutations. So these had a very high burden, but no response. So I want to ask, what can we actually decipher what's causing this high level of high neoantigen burden, but particularly high subclonal neoantigen burden? So we looked at these patients, and I'm showing you, again, the plots across the genome and looking at all these subclonal mutations. So lots of these mutations present at less than one copy in some of these patients' tumours. And particularly here you see in the middle patient here, tumour, we see thousands of subclonal mutations. And initially we thought, well, perhaps this is a sequencing artifact. Maybe it's just they've got a low burden. But what we could do is, again, similar to what we did before looking at the smoking signatures, we could look at what's actually causing these mutations. We could look at the trinucleotide context of each mutation. When we did this, we could see that the clonal mutations were caused by UV light. They had a signature consistent with UV radiation. But the subclonal mutations, on the other hand, looked very different. And these, rather than being a sequencing artifact, we could actually link these to alkylating agents. And so this was actually caused by the patient's prior treatment because they were treated by temozolomide. So while the clonal early mutations are caused by UV light in these melanoma patients, the later mutations were actually caused by the treatment itself. And these patients, bear in mind, did not benefit from the immunotherapy. So these are alkylating agent induced, which I think raises some questions about the impact the prior treatment can have on inducing mutations and the impact that may then subsequently have on subsequent therapy. In certain cases, this may improve the therapy. In other cases, it may not. So finally, I'd like to talk about the final part of my title is immune escape in lung cancer. And many of these tumours we've seen, there seems to be some interaction between the cancer cell and the immune system. Indeed, they do benefit from immune checkpoint blockade, but how do they escape the immune system in the first place? Are there any mechanisms that we can decipher here? So very quickly, I'd like to go through one thing we've been looking at, and that relates in particular to antigen presentation. So as I said, to predict neoantigens, what we do is look for whether the neoantigen, the mutation containing the peptide containing the mutation, can be presented by the patient's own HLA molecules. So one thing we can check is whether the, as I said, to predict the neoantigen, whether the peptide binds the patient's own HLA molecule. And these HLA molecules are highly polymorphic. So the HLA alleles that you have that are maternally inherited are quite different from those that are paternally inherited, because each specific HLA has very specific binding properties. And normally you would inherit two different copies of HLA A, B, and C from the maternal and paternal. And so each one will present potentially peptides and thereby neoantigens. And so therefore, if we look at the maternal allele and the peptides it presents, those will potentially be different from the peptides presented by the maternal allele. So there's some evidence in the literature that loss of a HLA allele can be beneficial for the tumour. Indeed, in a study that was looking at targeting the KRAS G12 neoantigen, which bound to the HLA-C allele, they found that upon relapse, the patient had lost the HLA-C allele, suggesting that this could be responsible for the relapse. So we wanted to ask, is it possible to identify HLA-LOH from this loss of this HLA allele from sequencing data? If it is possible, how frequently does it occur? And then when does it occur during tumor evolution? And ultimately, what is its functional impact? So why would it be hard to determine LOH at the HLA locus? Well, as I said, to look for LOH, we need heterozygous positions. We need those that differ between maternal and paternal alleles. And we need coverage at these positions. And the highly polymorphic nature of the HLA means that the standard reference genome is not very good, because the HLAs that this from a given patient's tumor may be quite different from the HLA reference. So the HLA reference, so if you use a reference genome, then this could be very different from the actual HLA alleles that patient harbors, because it's so polymorphic. So then when you align sequencing reads to the standard HLA reference, essentially these don't align. So this means that you get very poor coverage at the HLA gene. So what we reasoned was, instead of aligning to the standard reference, what we could do is we could align to the patient-specific HLA alleles. And so this is exactly what we developed a tool to do. We call this tool LOLA, which stands for loss of heterozygosity in human leukocyte antigen. So here we align the normal reads as well as the tumor reads to the patient-specific HLA alleles, and this allows us to determine for each patient-specific HLA allele the copy number within the tumor. So just to show you an example here, we can look in one patient and we can see, in this case, that the HLA A0101 allele is present at 2 copies, where the other allele is present at only 0 copies. So there's loss of an HLA allele. So we've got a tool now that we can work out copy number of HLA alleles, and then we can ask, how prevalent is this? Do we actually see this mechanism of escape in lung cancer? Or we could go back to the TRACER-X study and ask this exact question. And then looking at the 100 cohort, we actually saw strikingly that HLA-LOH, this immune escape mechanism, occurred in 40% of the samples. And this is particularly striking if we consider that mutations, single point mutations to the HLA genes, actually we see happening in less than 5%. And interestingly, we also saw this as differences between lung adeno and lung squamous cell carcinoma. With lung squamous cell carcinomas, which generally harbor more mutations and are heavier smokers, seem to have an enrichment for this LOH event. So we could next ask, well, when does this LOH event occur during tumor evolution? Is this an early event on the trunk of the tumor's phylogenetic tree, or is this a later event on the branches of the tumor's phylogenetic tree? And here, what I'm showing you is the different trees. And here we can see, we sometimes see it on the trunk. However, predominantly, we actually see it as a branched event occurring in a subset of the cancer cells and a subset of the tumour regions. And interestingly, in this case, we could also see evidence of parallel evolution occurring independently multiple times during tumour evolution. Again, suggesting a strong selection pressure here for it. And consistent with that, when we looked in paired biopsies where we had primary tumour as well as metastasis, we saw actually there seemed to be some enrichment for this LOH event to occur in the metastatic samples compared to the primary tumours when it was subclonal in nature. And finally, we could ask, what is the functional impact of this HLA-LOH event? Is it actually important? Well, given that it's selected, we'd assume it is, but what actually happens? Well, we predict that losing an HLA allele may diminish the capacity to present neoantigens. So if you've lost this HLA allele, these neoantigens may no longer be recognized by the T cell as foreign because they're not actually being presented anymore. So therefore, we predict perhaps if you have an HLA event, you might get more mutations because these are now silent and the tumor and the CD8 T cells, the immune system can no longer see them. So to look at this, we compared sister subclones that evolved from the same descendant to see whether when we have the HLA event in one subclone and it's not present in the other subclone, do we see more mutations where there's an HLA event present? When we did this, we saw actually a very clear trend for more mutations in the clones with the LOH event compared to their sister subclones without. And we could also see this separately in lung adeno and lung squamous cell carcinoma. And then also one interesting thing we could also see is when we predict these neoantigens, whether they bind to the lost or the kept allele, we saw a tendency for these, in particular subclonal neoantigens, to be binding to the lost allele that are no longer present, suggesting these are no longer potentially neoantigens. And overall, when we looked across the cohort, we could predict how many of these neoantigens that were predicted are potentially no longer neoantigens because of this LOH event. We could see in certain patients' tumours, in one patient in particular, 1,000 neoantigens that we predicted were no longer potentially being presented. And this represented over 90% of the predicted neoantigens. I think this suggests something we need to take into account. We're trying to design vaccines, therapies that are exploiting neoantigens. And finally, we could also look at the immune microenvironment of tumours with an HLA-LOH event. And here we could look for staining of PD-L1, so immune checkpoint marker here. And what we could see here is when we had a clonal LOH event, we saw far greater staining for PD-L1 on immune cells as well as tumour cells, suggesting potentially this LOH event is clearly associated with an active immune microenvironment, whether this occurs therefore in response to an active immune environment, or whether you're seeing the LOH event leading to somehow immune infiltration as well, we're not entirely clear. But you'd assume potentially this is the case, that you have an active immune environment, then you get selection for HLA-LOH, and those are the cells that survive. And we could also look at RNA-seq data for this subset to calculate immune infiltrate measures, and here again we could see that we see an association, particularly in lung adenocarcinoma in this case. When we have HLA-LOH, we see a more active immune environment. So that leads me to conclude what I hope I've shown you today, is that we think tumour heterogeneity is widespread in early stage non-small cell lung cancer. So we see this both at the mutational level and also the copy number level. And we think this copy number heterogeneity indicates dynamic chromosomal instability. And it's the copy number and not the mutational heterogeneity, interestingly, that is associated with reduced relapse-free survival time. And in addition, one thing we see very frequently in these tumours is a whole genome doubling event. We see this is associated with intra-tumour heterogeneity, and we think this may facilitate cancer genome evolution. And finally, after the doubling event, one thing we can see is ApoBec-mediated mutagenesis. The enzyme involved in innate immunity appears to be mutating the cancer cells themselves, and perhaps facilitates also branch tumor evolution and subclonal expansions. And finally, we also see, in terms of relation to the immune system, that clonal neoantigens appear to hold prognostic relevance, both in the context of patients treated with immune checkpoint blockade and even those that haven't been treated. And we also see a potential mechanism of immune escape that's particularly pervasive here is loss of your HLA alleles, so you no longer present potentially neoantigens. So that leaves me to thank everyone that's been involved in this work. This is really team science and tracer activity led by Charles Swanton, and also set up by Mariam Jamal Hanjani and a whole host of people that have been involved in this work, in particular Rachel Rosenthal, involved in the neoantigen HLA loss, and then everyone else also that I'm afraid I've neglected to mention during this talk.
Thank you, Dr. McGranahan, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Let's get started. Our first question is, do you think intra-tumour heterogeneity at mutational level is not important?
So what I'd say in response to that, I think clearly we see that copy number heterogeneity is important in terms of prognosis, but I wouldn't say that suggests that mutational level heterogeneity isn't important. I think we probably need to develop more sophisticated measures of heterogeneity at the mutational level, and indeed we do see the number of clonal neoantigens seems to be prognostic. So I think there's plenty more work to be done in that field.
Thank you. Next one is, do subclonal drivers exist?
So I guess this is something that I didn't focus on too much during the talk, is whether or not the subclonal expansions, what's driving those, and overall, if we look at most known drivers, these tend to be clonal early events. So that then leads to the question about what about the later events? I think again, this is an area that as we get more data, we'll get better at answering these questions. I think there often probably are clear drivers of later expansions, and sometimes we can annotate these and we can tell what these are, but in many cases we don't yet know what's causing the later events during tumor evolution.
Interesting. Thank you. We have time for one more question. How many tumor regions do you need to sample to fully capture the extent of heterogeneity in a tumor? So I think that's a pretty tricky question, actually, to know, to fully grasp the extent of heterogeneity within a tumor. And I think ultimately every cell within a tumor will be different from every other cell. So I think we could go into as detailed as possible, but then the question really is what is functional and what isn't. So what's important heterogeneity and what isn't. And there I think unfortunately, again, it's a case that probably varies from tumor to tumor. Some tumors, there's more very important heterogeneity, and other tumors, the most important events probably occur early on. So again, I think it's probably a question as we start to amass more data and start to understand these tumors, we might be able to start to get an idea of the evolutionary rule book of what events are very important early and what events are important later, and really how deep do we need to go. Thank you. I would like to once again thank Dr. McGranahan for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 11, 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That is all for now. Thank you for joining us. We hope to see you again